Good afternoon, everyone. My name is Dervla Morris. I am Director of the Ryan Institute Centre for One Health at the University of Galway, and it is my great pleasure to welcome you to our annual conference on today, World One Health Day. The Centre for One Health brings together researchers across all 5 colleges within the university with key external partners from the human health, animal health and environment sectors. We have 4 focus areas, including infectious disease and zoonoses, antimicrobial resistance, emerging pollutants, and people, nature, and the environment, with a cross-cutting theme of behavioural and societal change. Throughout this afternoon and tomorrow, you will have the opportunity to hear about ongoing research in each of these areas here at the University of Galway and from national and international experts in the field. If you have any questions for any of the speakers in each of the 4 focus sessions, please push them in the Q&A box. But first, it is my great pleasure to introduce you to our keynote speaker who will open the conference. Dr. Kasturi Venkateswaran, or Venkat, is the Senior Research Scientist at NASA's Jet Propulsion Laboratory and supports the Biotechnology and Planetary Protection Group. He has over 40 years' experience of research in the field of marine, food, and environmental microbiology. He's applied his research in molecular microbial analysis to better understand the ecological aspects of microorganisms. He is leading the International Space Station Microbial Observatory projects to measure microorganisms associated with ISS surfaces and air. His research into the study of nutrient-deprived oligotrophic environment using state-of-the-art molecular analysis, coupled with nucleic acid next-generation sequencing technology, will allow accurate interpretation of data and implementation of planetary protection policies of present missions, helping to set standards for future life detection missions. Unfortunately, Venkat could not join us live today, but he has provided a pre-recorded presentation entitled Microbes in Space, Challenges and Potential Solutions for the Omics in Space, which we will now stream.
Okay, some thought today, why microbiology in NASA? Everybody thinks it is an engineering institution. What the hell we are doing there, right? But think about it. The scientists are the one who drive the whole institute, whole agency. You have to ask the right thing. In here, we are asking, are we alone? Did we come from somewhere? Did we get implanted here? So if there is a case for one case is Mars, we might have exchanged the meteorite and then started with 3.7 billion years back. That molecule get adapted to the conditions of the Earth and then flourished from the primitive single cell bacteria to human we are. Okay, so we can ask that question. To do that, that is the science. The biology is the main thing here that drive the 99% of the engineering course, fact. Obviously, the one that we landed in Mars is the engineering fact of the century. 20th century is engineering feat of the 20th century. Astrobiology, I already said, if you are sending a mission to look life in other planet, that is first you do the robotic mission. So robotic mission, if you're looking for life better, you clean yourself. Otherwise, you do not know what you are measuring. That one I said the planet and planetary protection. That is also robotic mission. Then you look for life and you should not contaminate now. If you contaminate, whether the red planet, which you are seeing right now in Mars, may become green otherwise tomorrow, if you are not taking care of it. Same thing here also. When you go to the national park and other things, take your own garbage along with you, right? Same thing, that we should not contaminate our planet. Now, after these robotic missions, we are trying to go, the human are trying to go there. So for that, how the microbe differ, how that changes, whether there is positive or negative influence towards the human. Here, that's what the human research program, that whether it affects the crew health. The other one is you cannot take everything with you if you are going to go and explore other planet. If you want to have a sustainable life in Mars, another planet, better take the best out of the microbes with you, even making oxygen, removing carbon dioxide, anything that making food. Okay, so everything that you think of, for a sustainable life, you need microbes. So while doing that, we found several novel microbes that are, producing something, and that I will touch something. And then unusual microbes characteristics will be helpful. For example, if it forms biofilm, how you are going to remove it. And the whole genome sequencing will help us to understand those characteristics. And then proteomes, metabolomes, metagenome to phenome, the things that you do not know how to culture. You said emerging disease, emerging pathogen, right? So that one, we can come back from the metagenome, from the genome-based analysis to culture them. Once you have in culture, then you can. Okay, next one is space station. This is what I'm my sampling coming through. Anybody knows this guy? He's Scott Kelly. He's the one who spent in space station for more than a year. I mean, almost a year. This is the guy, Captain Terry Wirth. He's the one who collected our samples. And I had about 9 years of sample collection from space station. Astronauts can live maximum 6 months. Some people extraordinary like him are living 1 year, but maximum is 6 months. So every 6 months you have the crew changes. So in 9 years, I have multiple crews. So that means what? The collection people, either they are efficient sometimes, they are not efficient sometimes. They may shed their microbiome. All these things are also varied. So we have a baseline study also. This one is a video, but I'm not going to show all the 9 minutes of this video, but some of the things I'm telling here, we monitor how they collect the samples. Okay, so here he put the samples to collect the air samples. This is air sampler, and then you put an appropriate filter there, and then collect. So he comes and put it a kind of air filter. It's not a petri dish, but it's the same size. He put it. Look at it. He's not wearing the glove. So we are monitoring, and then telling him, "Hey, you are doing this not correct. Let's wear the glove." And then, how difficult wearing the glove? You guys are very good in when you are doing the surgery and other things. You are changing your glove, but still here, but your mouth and other things are covered, and you should not touch, right? So somebody is helping here. It's not happening there. He's struggling to have the sterile glove, that is different from normal glove, and it is floating in the because of the lack of the gravity. Okay, so he has to struggle in putting those things. So that kind of thing. So after that, he get the samples, wipe the samples appropriately. Now he done the gloves, and then he's taking the wipe, because he's touching the wipe, because we don't have a better sampling collection device that collects 1 meter square, efficiently. So he's collecting these kind of things that we are giving it to him. See, a lot of things these are Velcro, sitting on his various part of his body. And that's how we are collecting. Still, it is not a sophisticated sample collection, but we are collecting some samples that will be coming back. While doing so, what happened? Sometimes the following thing happened. Somebody swing by you. That means when you are doing a surgery, if somebody is not clean, that person will contaminate. So that's what is happening here. And we are collecting. While we are collecting the people who are inhabiting in the environment. Sometimes, it is not only for ladies, but anybody will chatter things next to you. If they want to say something to you, they'll come and then they do some experiment next to you. Okay, they are non-stop. They are not wearing the there is no corona there, so they don't wear mask there, and but they speak. So his oral microbiome, her microbiome will also be there. So all those things are there. I'm coming to this, because I have to go faster here. This is the one I'm going to talk today. First of all, as I said, challenges, biofilm, how you can develop certain, antimicrobial coating. That helps. Otherwise, biodeterioration. 10 years back, you might have built a house. Now you are seeing some problems there. You can easily change, but that's not going to happen in space station. Space station is 22 years. Either you abandon them, destroy them, which was, more than $200 billion worth of stuff there, or come up with a technology to develop something new, different. And the other one I also told, because you guys are a medical professional, you need to know the problems and associated things. So in space station, microbes develop more antimicrobial property, more virulence property. So you need to come up with some sort of antimicrobial thing. And also it produces some virulence factors, right? So that resistance is the biggest thing. So I will tell a Kalamiel appears on his story. As you know, this is named after Abdul Kalam, a genus, okay? And then we also need appropriate instrumentation to do, you are sending your samples for sequencing. You don't have a FedEx in space station to bring it down and then do things. You have to do everything there. How many moving parts are there? How do you collect, even transfer the fluids from one to the other, right? So those are the things are there that we need to work on. So I'm not going to cover all these things that I worked there around 25 to 27 years now. This is Elon Musk standing next to his SpaceX. So that is the one is transporting things to space station. So that means it is there is one source, right? And the other one is, this is Earth analog. Crew time is very hard to come by. 1 hour of crew cost about $2 million. Astronauts are not getting the money, but you are sending things, all other things. If you calculate things, each experiment that I'm sending it for space station, it should be simple. It should not consist of astronauts' time. At a given time, 160 experiments are going side by side. Astronauts, only 6 people. They may not be microbiologists. They may be engineers. So they can do switch on, switch off. That's it. That is his microbiology experience. That means you have to bring such a kind of simple instrumentation to space station. Okay. Anyway, so first you need to know what is the baseline there. That's why the inside is space station, and the other one is things in space. What is happening? All other things are I'm not going to talk today. But anybody interested, please talk to me through Dr. Damodaran, and we can do that also. So these two I will try to cover. We came long way. These are nothing but a simple petri dish, grow them. All of you might have done the microbiology, to the sophisticated metagenome as we know today for ultra clean biomass environment. Whatever we are doing is drawing the blood out of the rock. That means very, very, very low level, below detection limit actually. Okay. So what we do, we collect samples as I told you, I showed you, okay? And then we also don't forget the traditional microbiology. Without that, you cannot develop countermeasures at all. Without that, you cannot come up with some sort of requirements to place. Everybody say, I'm having a countermeasure development already, I am killing the bacteria, leaving behind the DNA, I don't care, right? That is not going to do anything. But if you have the microbes, whether you are leaving behind or not, you will know, right? And also we develop technology how to selectively remove the DNA and other things as a remnant, only look for viable microbes. For all these things, you have to, the other one is biofilm, right? You have a spacesuit, but you don't have so much to pick. Astronauts come 6 months after that, he or she cannot have her own stuff. It is there, S, L, M kind of thing that you pick your size and then use it. What that mean? You are leaving behind something and the other person is wearing it, right? You don't have a laundry there. You have some chemicals to destroy it for sure, but it is not 100% sure. This is the texture is the spacecraft spacesuit material. Okay, and then this is the biofilm forming the red one, or the rhodotural mucilagiноsa, that actually making this biofilm on the spacesuit. But as I said, this is rhodotural mucilagiноsa. This is having two different kinds. One is clean, round. The other one is spiky stuff. Those spiky stuff are the one producing this thing. So not all the things that some subsample is doing. We need to develop some countermeasure. Here is the one that actually kills this kind of bacteria, and that you coat on the surface. This is stainless steel. Medical institutions also work with the different materials to look for it, right? So this is a water processor materials. 5 materials we tested all killed the Pseudomonas aeruginosa, no problem. But rhodotural mucilagiноsa, that is not get killed. Why I'm saying is not one size fit all. We have to have multiple things. In this case, this is what is happening. If your surface coated with something and bacteria come in contact with that, yes, it get killed, including yeast or something. But when you have the detritus material there, the other one can sit on top of it. Now it is not in contact with the material, right? So that is what is happening. This is bigger, this is not that bigger. So here get killed, but here it is not. Okay, this is for stainless steel. This is for Teflon. You have medical devices with Teflon as well, right? So it is okay and get killed, all the Pseudomonas, but not rhodotural. This is, this is inconel, this is titanium, and mainly because of this thing. So this gets settled, and that is where the things get killed, but the other things are sitting on top of it. That's why there we started seeing so many novel bacteria. This is the one that I worked with the MTCC people. Mild Raj is my collaborator there, and then Solibacillus calamiae. This you are not when you are looking for all these things, you are not going to name each and every bacteria. This has to do something. It should have some characteristics. It should be problematic or it should be helpful. Okay, so Solibacillus calamiae is as you know, this is after my virtual mentor, everybody's mentor he is, and this producing exosporium. Some of you may be working on Bacillus anthracis, so on Sirius. So that has this additional layer. This is called exosporium. You can strip that off, but still it will be alive. But that help to get protected from radiation. This is made up of glycoprotein, and that can be used as a sunscreen. So that can be developed for astronauts. Spacesuit get coated, astronaut visors get coated, and also it is biotechnologically helpful for the Mother Earth as a sunscreen development. Okay. Then another thing, Methylobacterium ajmaliae. Damu knows. He is my senior, Dr. Ajmal Khan, and I named after him. And why I am characterizing this, this is a rod-shaped gram-negative bacterium. It helps to produce a hormone that promotes the plant growth. As I said, you cannot take your tons of soil with you to grow something. So you have to use moon soil or Mars soil to see whether anything growing. For that, you feed this hormonal stuff. This may be helpful for the soil to get fertile. You may be asking me, hey, first go and make Sahara Desert to green. Yeah, we are also testing that. Whether it is going to be useful for our Mother Earth or not. So we are using that too. So I named this after my friend, and he's not my immediate senior. I mean, he's my immediate senior. It's not. He's fish microbiologist, marine microbiologist. I am marine. See, he's marine biologist. I am marine microbiologist. we communicate and talk more the thing, but he's as simple as Abdul Kalam, very, very simplistic man. If you're simple, you cannot come up into your administration, but he's a very good scientist. That's why I named after him. The other one is chloramphenicol. So most of you prescribe chloramphenicol as a generic drug without knowing what bacteria it is. Some bacteria is actually growing up with that. Okay, I'm saying growing. It is not withstanding. It is growing. You put it in the presence of the things at appropriate microgram level, that we use it to contain bacterial growth when you're looking for fungal growth, right? So all of a sudden, some of the time you are getting this actually also producing biofilm. And I named after Kate Rubins. She is the astronaut who sampled my things from space station, and also she's the first human who sequenced DNA in space. She's a molecular biologist from Stanford as well. Okay, so now then it is not only bacteria around, now fungi. Fungi is also producing biofilm. So these are the forms of these carton-like structures that you are seeing. What you are seeing in the middle are those, conidial spore. Okay, so these things are these are hyphae, fungal hyphae. And this was novel, isolated from Mars 2020 assembly facility. I named after one of my great colleagues. He's the one who collected all the Chernobyl fungi. He's a mycobiologist. So Thomas Torok, so I named after him, Torokii. And then another example for yeast. So you saw bacteria, you saw a fungi, you are seeing a yeast. Anybody working on Cryptococcus here, neoformans? So you medical professionals may know Cryptococcus neoformans is a deadly yeast infection. One of the characteristics is nothing but the Titan-like cells. It forms balloons when it gets into your system. Okay, so this is the first novel eukaryote isolated from space station. I named after a person, philanthropist, who spent money to do the research to Chris Mason, actually, Damu knows, and his name is Igor Tulchinsky. He spent $5 million for this, so for the space station sequencing project. So we named after him. Here is the thing, what I said. Here is a normal yeast cell, which all of you who are working on yeast may know, under 1G. 1G means normal gravity. Okay, and if you are growing it into the 5% CO2, which is the human thing that you can mimic to some extent, it is still the same, a little bit bigger, but it's still the same. But when you grow the double punch, microgravity plus CO2, it balloons. This is the kind of things happens for Cryptococcus neoformans when it become pathogen. We are still working on this. Multi-omics analysis is being carried out right now, and we are still working on this. Okay, so one other thing that is very interesting is yeah, these are all there. It's not a rare event. People may ask, it may be there because it can happen in your home. It can happen in your hospital. Why do you need to worry? Persistent is the biggest issue, right? So let's take the BSL-2, all of them. So now you are not able to see everything, but some of the one that is bigger font are related to the incidence of it. That means it is there in numbers. That's one. Is it one time? That's another thing, right? So you can say, it is one time, I'm cleaning, it is gone. If it persists, that is what is happening here. These are flight 1 to 3. Flight 1 to flight 2, 3 months difference. Flight 2 and flight 3, we planned 3 months difference, but SpaceX 6 blown away. NASA had no vehicle to go there, so 1 year. So flight 1 to flight 2 is 3 months, and flight 2 to flight 3 1 year. So that means it is still persisting, and third year it actually spread to other locations. These are the locations of the space station, exact same locations. We went there to collect the samples that I showed you before, the astronauts collected. And then the other one is is that initial 2 different strains same or different? For some reason, this here I'm not seeing anything. Other Staphylococcus aureus popped up rather than Klebsiella pneumoniae. As you guys know, it is a pulmonary infection, Klebsiella pneumoniae. You know, if you have a corona plus Klebsiella, it is a big problem, right? So is the spread of this species, which is an opportunistic pathogen coming from one strain. Klebsiella pneumoniae, you guys know it is an opportunistic pathogen, it is well treated, but can you treat in an immunocompromised person? That's a big deal. Astronauts are very, very, very healthy, but when they be there for 6 months, psychologically disturbed, physiologically compromised, because they don't know where the head, where the toe is. They have the fluid exchanges from top to head. Right now we have a different. How many of us can stand upside down even 5 minutes? Right? So they are there for 6 months. So they are immunocompromised. A benign microbe, even a simple E. coli, may be a problem. So we need to come up, how do you find? We have the sophisticated technology like metagenome analysis to be carried out, and then we have the data in a public database to mine. And is this by one single strain? If so, how do you eradicate? Gene therapy, like phage therapy. If you come up with the appropriate phage that actually put that, then now you can mix that with that. So now for that, you need a basic science to understand. So one of the things that is metabolic modeling, how this Klebsiella is helping, or how do we kill it? Now let's see how it is helping to produce some antifungal compound. So you can do the metabolic modeling of all these things, and then this Klebsiella is acting upon. This is parasitism. That means if you have this mark, is parasitism. This is mutualism. Commensalism means mutualism. Okay, so here the rhodotorula are actually killing Klebsiella. Rhodotorula are killing Klebsiella, but Klebsiella is killing Aspergillus penicillium. You can predict this. Is that right? You have to validate it, because we have skepticals sitting in front of us saying, I am experienced, you tell me how you can prove this, right? Okay, this is the fungi, Aspergillus fumigatus. All of you know BSL-2, pulmonary infection. Again, it is an airborne thing, okay? This is hyphae, and these are the conidial spores that is very healthy under the regular condition. For the past couple of decades, microbial monitoring is happening based on omics technology.
Omics includes everything inside the microorganism, not just their species identification through their DNA, but also what enzymes they're producing, what proteins they're producing, and what metabolics they're doing. And as we study microbes in space, these are key things to take into account, because those can give us clues and hints as to how microbes are changing and adapting to the space environment.
So currently on the International Space Station, there's no mechanism to be able to process samples. The process for microbial monitoring on the International Space Station is for astronauts to collect samples from either the surfaces or the air with swabs. Once the samples are ready to be returned to Earth, it gets put into a capsule and then it lands into the Pacific Ocean. A boat then comes to collect the samples, which is then transferred back to different NASA centers. So it's very important if we could cut all of these time and these losses by performing sample processing on the International Space Station itself.
So one of the things is we need an automated sample processing instrument, which we call them as SPI system, that will be helpful to measure the microbial population in space in a closed system during the long travel.
The SPI is a microgravity optimized DNA extraction device that's ideal for use on the ISS. The crew time will only spend about 1 to 2 minutes per sample, and then they could use our device to process multiple samples simultaneously, which means the data that they collect eventually will be more reliable, simply because more sample and more repeats can be performed with our device.
So once an astronaut collects the samples, it would take only a day for us to get results back rather than about 4 months, which is now the current protocol.
But we do not know whether it is going to work in space, because fluids behave differently under microgravity.
The SPI device can successfully extract
I'm not sure what the issue was with the audio, but maybe we can come back to that presentation at the end of this session. Dervla, if you're there to take over here.
Yeah, thanks. Apologies, everybody. There seems to be some technical issue with the audio coming across there, so sincere apologies to everybody for that disruption. In the interests of time, we'll try and resolve the issue with the audio, but apologies if we cannot go back and play the end of that presentation. We will now move directly to our next speakers. Our next speakers are Dara Harrington and Esmeralda Cecile Perez, both of whom are fourth-year medical students here at University of Galway. Dara is Secretary of the Irish Doctors for the Environment Student Group in Galway, and Esmeralda is the student lead for the Education Working Group in Irish Doctors for the Environment, leading the student call for increased planetary health education in Ireland. In 2020, she was one of the key contributors in implementing the Planetary Health Report Card in Ireland for the first time. And in 2022, she co-authored a report on the primary Planetary Health Report Card findings, which was published in Medical Teacher. Dara and Esmeralda will present a presentation entitled Planetary Health Report Card, a Global Student Initiative. So over to you, Esmeralda and Dara.
Thank you very much. I'm just gonna share my presentation. So can somebody just confirm that you're able to see the slides?
Yes, we can see it. If you want to put it in slideshow mode.
Yeah. No. Just a second. There. Okay.
Great, thank you.
So hi everyone, I'm Dara and I'm joined by Esmeralda. As Prof. Morris introduced, we're fourth-year medical students here in the University of Galway, and we're representing Irish Doctors for the Environment, and we're going to be talking about the Planetary Health Report Card, which is a global student initiative. Next slide, please. So just as an outline, we're going to be talking about IDE and our work. I'm going to touch briefly here upon the planetary health concept, and then Esmeralda is going to talk more about the Planetary Health Report Card, its methodology and our findings and how that relates to the University of Galway. So the definition we're working with for planetary health is the one used in 2015 when the concept was first launched in medical literature. It defines planetary health as the health of human civilization and the state of the natural systems on which it depends. So as you can see from this diagram here, there's significant overlap with One Health, and you can sort of think of planetary health as an extension of public health. It deals with human health as seen in the context of external systems, such as societal structures like economics and environmental structures. It places emphasis on the effects of anthropogenic climate change, pollution, and loss of biodiversity on health, viewing these as some of the main threats to planetary health. Next slide, please. Thanks. So Irish Doctors for the Environment, what do I mean when I'm talking about this? Well, it's an NGO consisting of doctors and healthcare professionals, which has the goals of raising awareness, increasing interest, and taking action around the links between health, environment, and climate change. And I'm just going to talk a bit about some of our projects. Next slide, please. So as you can see here, these are some of the activities that the NGO is involved in. They organize events, such as the one pictured here in Dublin, and demonstrations. They're involved in research, for example, a recent publication here on the inhaled anesthetic agents in Ireland and their carbon footprint, and activism and planetary health advocacy, as well as general environmental advocacy in Ireland, Pictured here is a recent letter to the Irish Medical Council and the Irish medical schools calling for clarity on planetary health teaching in medical curricula. The way Irish Doctors for the Environment is structured is that it's subdivided into several working groups, and we're both part of the student group, which traditionally has focused on planetary health in the curriculum and changing medical curricula across the country. Next slide, please. So this is just a brief overview of some of the projects that our student group has been working on. You can see here, this is a summary of the Planetary Health Report Card from the first year that what formerly was NUI Galway was involved in 2021. And off the back of that, we were able to get a publication in Medical Teacher based on the results. We host awareness events and information talks, such as one this time last year, which was aimed at both at students, healthcare workers, and faculty within the university. And this was a really successful event. We had guest speakers from faculty and healthcare workers involved too, which is great. And off the back of a lot of these projects and achievements, we received an invitation to join the Global Consortium on Climate and Health Education. This invite was extended to the School of Medicine here at the university, and they're currently reviewing this invitation. And these two images here are Esmeralda, our student group lead. She has given presentations as part of the European Public Health Conference and talks with the Climate Alarm Clock podcast. So why is it that we're so concerned with planetary health specifically in medical curriculum? You might be wondering what the link sort of there is. So the World Health Organization in 2021 states that climate change is the single biggest threat to humanity. But despite this, a survey conducted by the International Federation for Medical Students Association found that only 15% of medical schools worldwide actually have teaching on climate change at all. So this was something we wanted to provide more research around, and that sort of led to how our group became involved with the Planetary Health Report Card. Next slide, please. So Esmeralda is going to talk a bit more now about the report card itself and its findings.
Thank you very much, Dara, for taking us through the background and who we represent, so the Irish Doctors for the Environment. And I'll just go through a little bit about the Planetary Health Report Card, which is the biggest project that we do here at the University of Galway. So the Planetary Health Report Card is a student-led initiative to assess how much medical schools engage with planetary health. And it was originally started in the US in 2019, but has since become expanded to the rest of the world. So you see a map here showing all the schools that are currently involved. You see there is a lot of focus in the US where it was started. Now the UK and Ireland, as well as Germany from Europe, are involved. And we also have a few schools in Asia involved. And we're hoping to expand this further as we go on. And since it was only started in 2019, you can see how much it's expanded in really only 3 years, and it's become a global health movement. So you see here in the top as well that it kind of gives an overall grade of the different schools, and you can see that there's a lot of orange and red, so C and D, which kind of reflects what Dara was saying, that there aren't a lot of schools that are teaching about climate change or the links between environment and health sufficiently. But we're hoping that this will improve as we keep doing this Planetary Health Report Card to push for more planetary health teaching. So the way that we assess the different medical schools is based on 5 different categories, and they are the curriculum. So we're looking at the curriculum, we're going through the curricular plans, we're talking to lecturers, we're talking to students who have gone through the curriculum, and we're just looking at how much planetary health teaching is there, how much are they bringing up this aspect that climate change is a threat to health, how much are they bringing in the aspect of the link between environment and health, and so on, and then we grade them in that category. The second category is interdisciplinary research. So how much is the School of Medicine engaging with the research centres that are present at the university? Are they trying to create research opportunities for students to get involved in planetary health or One Health research? And then for the community outreach and advocacy, we're looking at how much is the School of Medicine engaging in planetary health, trying to get the message out to the general public? Do they host conferences? Do they host public talks? Or do they have any other initiatives to try and create awareness in the public? We also look for the support for student-led initiatives. So how well are they receiving our initiatives? Are they receptive to what we're trying to achieve? And are they trying to accommodate us as a group? Are they trying to meet us, for example, by having regular meetings with us and such things? And then the last one is campus sustainability. So we're looking at how sustainable is our campus, and this is more of a university-wide approach, it's not just the School of Medicine, but are they trying to divest from carbon fuel? Are they trying to upgrade buildings so that they're more energy efficient and so on? So now I'll talk you through a bit about the methodology, because how exactly are we grading the schools in all of these different areas? And what we're using are metrics. So the picture here to your left, that's an example of a metric that's taken from the first category, so the curriculum, and it's the 9th question in that category. So each metric starts off with a question, and in this example, it's, does your medical school curriculum address the unequal health impacts of climate change globally? And then you can see that you get points based on how well they meet this criteria. So 3 points, which is the best if it was explored in depth by the core curriculum, 2 points if it was only briefly covered in the core curriculum, 1 point if it was covered in elective coursework, and 0 points if it was not covered at all. So based on this then, based on going through the curricular plans, talking to faculty, talking to students, we get an idea of how well this was covered, and we give them a score. So when we have a score for one question or for an entire category, we accumulate the scores, and in that way we can get a percentage. For example, say that the max score that you can get in the curriculum category is 69, and by going through the metrics, you got 20 points in total. So 20 out of 69 is 29. And then looking at this kind of box here, it tells you what that percentage equals to a grade. So in this example, with 79%, that would be a grade of D, and we would give a grade of D for the curriculum category. And we go through all of the categories in this way, give them a grade. And then when we have a grade for all the categories, we can also give an average grade for the whole school. So this is the results from last year's, or from 2022, which was released this March for Ireland. So as you can see, we have the schools here. So that's all of the undergraduate medical schools in Ireland. And then on your right side, we have the scores, or the different grades. So you see University of Galway, formerly known as NUIG, we got an overall score of C, which is not the best, but not the worst either. And it's pretty consistent with the other schools here in Ireland. We got a D in the curriculum, and it was mainly because by going through it and from our own experiences and talking to faculty, there really isn't that much of planetary health focus in our curriculum, sadly. However, what we did find is that there are a few lectures that are given actually by the Centre for One Health relating to zoonoses and relating to some environmental risk factors for health, which kind of boosted us from an F to a D. So what we would really like to see is if this could be improved. And as the University of Galway is currently going through a curriculum change for the medical curriculum, there really is a great opportunity for the School of Medicine to include more planetary health focus. We do really well in research with a B, and this is again mainly because we have research centres such as the Centre for One Health that are actually delivering some lectures to the medical students. And yeah, we're doing really well with the B, but the only way that we could do better to get an A would be to enforce those links a bit further, try and get research opportunities for students, for example, in the Centre for One Health. These kind of things is what we would looking to improve that score even further. For community outreach and advocacy, this is where we score the poorest, and that's mainly because the School of Medicine isn't as involved in the community outreach. So there might be other initiatives from the university, but we're really grading the School of Medicine, and they aren't necessarily that involved in giving public lectures or other initiatives in the community outreach and advocacy. So this is something that, again, we would look to improve. And then for the support for student-led initiatives, we have found that the School of Medicine is really receptive to our group as representing the Irish Office for the Environment. and that they have met with us to try and see how we could try and improve our score in the planetary health report card. However, what we're kind of lacking a little bit is more of the structured approach where we could have a designated person that we could reach out to in the School of Medicine. Maybe if we could have regular meetings. And we're also lacking funding. It's one of the criteria for getting a better score in the support for student-led initiatives. And then for campus sustainability, we're also doing really well, mainly because the University of Galway, as a whole university, we're really trying to, they have a really good sustainability program or strategy. They're trying to divest from fossil fuels, and they're really doing a lot to revamp the buildings so that they're more energy efficient, a lot of recycling opportunities, and they recently had a project trying to increase pollinators on university grounds and all of these things. So that's really good, and we should just keep doing that. So this slide here is just for the trends at the University of Galway over the last years. So the first time we included the Planetary Health Report Card in Galway was in 2021, and then we have 2022, and we're doing it again for 2023. We're currently just in the process of starting that up again, and we're hoping to keep this going every year just to kind of keep tracking and seeing the trends over time. So there isn't really an awful lot of change between the 2 years. We see that we improved a little bit in the community outreach. We also improved a little bit in the support, and that was mainly because after the first Planetary Health Report Card was released, that kind of put us on the map, and we became a bit more known to the School of Medicine, which allowed them to include us more. And we've also been invited to participate in the curriculum review process, which is currently ongoing. So that's also what's kind of increased that a lot for this year. We've actually gone down a little bit in terms of the curriculum, and that's mainly because previously we used to have special study modules, which would be the elective coursework referred to as earlier, which in some of those special study modules, we could have chosen things which were a bit more related to planetary health, but because of COVID, a lot of them just disappeared, and we haven't really seen it come back either. So that put us back, unfortunately, a little bit for the curriculum overview. So based on these findings, we have a bunch of strengths, weaknesses, and opportunities, which we also have given to the School of Medicine formally in a report before. I'm just going to go through a few of them. So the main strength of the University of Galway really is that we have the Centre of One Health Research Centre. So a lot of the other universities in Ireland doesn't have access to such a research centre, and it really is a great strength for us to have it. We also, the university-wide campus sustainability effort really is a big plus to give us the scores that we have. Unfortunately though, the weaknesses is that we don't have any explicit learning outcomes on planetary health, but hopefully this can change now that we're going through the curriculum. There also is a bit of a poor community outreach effort by the School of Medicine, but because there are other efforts in other parts of the university, it would be really easy to just try and link in a bit more with the wider community and try to get involved, bring the health aspects when we're talking about environment or climate change in other scenarios or in other contexts. So for the opportunities then, the current curriculum review is a really good opportunity for the School of Medicine in increasing their planetary health focus. And by inviting our group to have a representative there, they really are taking it seriously, and we're hoping it will bring results when we finish the curriculum. Then another opportunity is to just link in further with the research centre we have, like I mentioned many times now, to just link in with the Centre for One Health even further, try to get the One Health aspect or the planetary health aspect into not just public health, but also into other aspects of our education would be great, or even to just have a research program, link in the Centre for One Health with the research program. We have a summer research program with the School of Medicine, to maybe link in the Centre for One Health with that summer research program would be great. And then really University of Galway, because of these things, they have a really good position to become a pioneer in planetary health in Ireland. So we're seeing some trends. The RCSI, they're really trying to revamp their curriculum, trying to become more planetary health focused. But because we're currently going through a curriculum review, and because we have the centre, University of Galway really have a unique position to try and lead the way in Ireland. So that brings me to the take-home messages. which really are that as future doctors, it is important that we as medical students have a sound knowledge of planetary health, precisely because of what Dara mentioned earlier, that it is the biggest threat to health worldwide. We aren't really taught about it. The second take-home message then is that there are students who are working to increase planetary health engagement in the Irish medical schools, such as the student networks in the Irish Doctors for the Environment and the students involved in the Planetary Health Report Card Initiative. And the last thing then is just to reiterate again that the University of Galway really is in a unique position to become a leader in planetary health through adapting its medical curriculum and linking in with the Centre of One Health and the Global Consortium on Climate Health Education. So that really brings us to the end of our presentation. I just want to say thank you on behalf of Dara and I, and just to extend an invitation to any faculty or student present. Like I said, we're currently doing the Planetary Health Report Card again, so please feel free to join our team if you're interested. And you have our emails here at the bottom. So thank you very much.
Thank you, Esmeralda. You can stop sharing your screen now, if that's okay. It was a fantastic presentation, and thanks to both all three of our speakers in this opening session. I think in particular it's fantastic to see our undergraduate students getting involved in such important initiatives, given that they are our future. So we will now move on, and again, before we move on, I want to apologise again for the technical difficulties we had with our opening presentation. It's beyond our control, unfortunately. Unfortunately, we aren't in a position either to go back and try to restream it. So we will now move on to the first of our 4 focus sessions. Our first focus session is focus on infectious diseases and zoonoses. And I will pass you over to the chair for that session, Dr. Carolina Verissimo. Carolina.
Hi, thank you, Deborah. So, as Deborah introduced me, I'm Carolina Verissimo, and today is my pleasure to chair the session of infectious diseases and zoonoses. And we have a few speakers there that reunion very interesting talks in very different areas of the infectious disease and zoonoses. So I think it will be very interesting to show the diversity of diseases and problems we have in this area. I would like to start by thank the 4 great speakers that we have that agreed to share their knowledge and work with us today. So thank you very much, Dr. Anthony O'Donoghue, Dr. Nicola Calvani, Dr. Rihanna Walsh, and Dr. Claire Kenny for agreeing to participate. I also wanted to ask the audience to not be shy and post the questions in the Q&A. We'll have a discussion at the final part of the session. Will be 25 minutes more or less, where we'll try to include all the speakers. So please put your questions there in the Q&A and direct the question to the speaker that you want, so I can organize better at the end this discussion. Also, I would like to just remind the audience and also the panelists that the session is recorded, although a few sessions after will be not available as requested by the presenters. So let's start by introducing our keynote speaker, Dr. Anthony. Dr. Anthony O'Donoghue is a professor at the School of Pharmacy and Pharmaceutical Sciences at the University of California in San Diego, US. And his talk today will focus on parasites, selective proteasome inhibitors, and anti-infective agents. Dr. O'Donoghue obtained his PhD from the University of Galway, and then he moved to working as a postdoc researcher at the University of California in San Francisco. And then he has joined the University of San Diego in 2015, where his research is focused on the development of protease inhibitors, activity-based diagnostics, and protease activated therapeutics. and he is the current president of the International Proteolysis Society. So thank you, Dr. O'Donoghue, and welcome back to Galway, even if you're virtually. I will pass on you there.
Great, thank you, Carolina. Can you hear me? Great, and you can see my screen?
Yes.
Fantastic. Let me see if I can get my pointer up here. Oops. There we go. So thank you for the introduction. So as Carolina mentioned, I'm at the Skaggs School of Pharmacy and Pharmaceutical Sciences here at UC San Diego on the west coast of the US. And this is some images here of our beautiful campus. We're right up on the ocean here on the Pacific Ocean. And this is the building I work at here, the School of Pharmacy building. I'm on the 4th floor of that. And that School of Pharmacy building is here. And our campus goes right out to the ocean where we have the Scripps Institute of Oceanography. And that will be a part of my talk later when I talk about my collaborations with the Institute of Oceanography. And so just a little bit about myself and my scientific journey from Galway to San Diego. I'm actually from Cork originally, from Cove. And if you've never been to Cove, you need to go down and check it out. This is some stock images I took of Cove, and my house is just here. So I've always been growing up by the water. And actually I was home a few years ago, right before COVID. And yeah, we get a lot of the cruise ships coming into Cove. And actually you can see that there's 2 little canoes there. This picture was caught on the Irish Examiner. And this is me and my brother sitting out here in the harbor checking out these cruise ships. So my journey began where I did my PhD, and then I went on to do I did my degree and my PhD at the University of Galway. And my PhD work was in fungal proteases, and this is where I got into this area, and I've continued to work for the past 20 years or so. I've continued to work in the field of proteases and still do some fungal protease work. But when I moved to the US, I initially ended up in San Francisco, and I did a postdoc working on proteases in cancer and proteases, viral proteases. And then I moved to San Diego, and in my lab here at San Diego, I work on proteases in infections, cancer, inflammation, wound healing, digestion, clotting, and neurodegenerative diseases. So anything to do with proteases, I tend to have some sort of involvement in it. But today I'm going to talk about one particular protease that plays a key role in infections, and that protease is called the proteasome. So this is essentially a protein degradation system that we have in all of our cells. So some protein that needs to get degraded first gets ubiquitinated. So this is some signaling in the cell for degradation. So the protein gets tagged with these ubiquitin peptide chains that connects it to the proteasome, and then essentially the proteasome unfolds this protein substrate. It feeds it down through the central core of the proteasome, and shorter peptides come out the end. And so if we kind of take a cut of this, what we can see is that this is a ATP driven process of unfolding. The ubiquitins are removed by enzymes called deubiquitinases. They go off and ubiquitinate some other protein. The unfolded protein then gets fed down, and here is where the proteolytic activity occurs in this red region here. And I've just redrawn this core particle here, this blue, red, and blue particle, and it essentially is composed of an alpha ring, which contains 7 different proteins, which are called alpha proteins, and then 7 different beta proteins, and then the same 7 are inverted here and here, so they're like a mirror image of each other. And if we take a cut across here, we can see that the protein that we call beta 1, beta 2, and beta 5 are actually catalytically active proteases. And I've drawn the little mouths here in different shapes because they cut proteins and peptides at different sites. So they have different, what we call substrate specificity. And so for human diseases, for cancer, there are 2 approved drugs. And these drugs essentially mimic peptides. So this is like a phenylalanine and leucine. So it's like the peptide is the bait for the proteasome that fits in the active site. And then you have the hook, which is a reactive electrophile that reacts with the catalytic residue. So we have our bait and our hook here, and these molecules fit in the active side. This is bortezomib, which we trade name of Velcade, and this is clofazimab, trade name of Kyprolis. And both of these compounds are approved drugs for treatment of multiple myelomas. And they work mostly by binding and inactivating the beta-5 subunit, and at higher concentrations, they'll also inhibit the beta-1 and the beta-2. So how does this kill a cancer cell? Well, if you can block proteasome degradation, the cell has already committed to unfolding a protein for degradation. But if you block the degradation part, you end up with a whole bunch of unfolded protein in the cell. And cells don't like unfolded protein because they're sticky, they bind to everything. And so the cell undergoes apoptosis if there's a high level of unfolded protein. So essentially, you can kill a cell by blocking the proteasome, causing an increase in these toxic unfolded proteins. Now, switching to parasites, so parasites also have a proteasome. I just changed the color here just to compare the parasite proteasome to the human proteasome, but essentially they have the same three catalytic subunits. And I work closely here at UC San Diego with parasitologists, and these are all of the parasites that are currently growing at the School of Pharmacy. So I have different collaborators that culture these parasites. This is the disease, this is the parasite, and I've just indicated here whether it's an amoeba, a protozoa, or a worm parasite. And I also have colleagues in the building next door at the School of Medicine that also grow Plasmodium falciparum malaria, which will be the focus of my talk, and also some of these other protozoan parasites. Now, we and others have shown that you can take these cancer drugs and you can kill every single one of these parasites with the cancer drug. However, since these are cancer drugs, they're quite toxic. Because they also can kill human cells. And so they could never be used or repurposed as anti-infective agents. So the goal of my talk today is to show you how we develop inhibitors that look like this, but are actually much more selective for these parasites than they are for the human proteasome. Okay. So one quick slide about the Plasmodium proteasome. So I'm sure everybody has heard of malaria. So Plasmodium falciparum is the causative agent. We won't go into the life cycle, but essentially some of this life cycle occurs in the mosquito. The mosquito takes a bite. The parasite enters the blood system and eventually enters the liver, where there's a replication cycle, and then it moves into the red blood cells, and there's another replication cycle. And colleagues of mine at Stanford, in particular this former graduate student, Hao Li, she had done a screen where she tested a whole bunch of peptide-like inhibitors. So these are peptides that look like the approved drugs. This is the bait here. This is the peptide sequence of 3 leucine residues. And then this is the hook of vinyl sulfone, which reacts to the active site of a proteasome. And what she found is that this compound was actually more potent at killing Plasmodium compared to human fibroblast cells. So she calculated the effective concentration to kill 50% of the population, which we call EC50, over a course of 2 hours. And the lower the number here, the more potent it is. So at 68 nanomolar, she was able to decrease half the population of Plasmodium, whereas it took 2.5 micromolar. So we had some nice selectivity of this of 38-fold if you divide this by this. And she found that this compound here inhibited the beta-5. But we asked the question, can we develop better Plasmodium inhibitors? And basically, can we get this selectivity number to increase by either decreasing this number or increasing this number? And so she reached out to me because at the time, while I was in San Francisco, I developed this peptide library for being able to characterize all types of proteases. And essentially, this peptide library is a whole bunch of 228 peptides that are each 14 residues in length. And I designed these peptides using some mathematical formulas, and that's described in a paper that we published about 10 years ago. I won't go into that detail there about the mathematics behind it. But what I can tell you is that all possible neighbor and near neighbor amino acid paired combinations are present within these peptides. So we have 228 different peptides. We add a protease to these peptides. The protease cuts these peptides somewhere along this chain, and then we can detect the cleavage products by mass spectrometry. So how do we use this assay, and how will we use this to develop better drugs? Well, we can take any protease of interest, mix it with, and we put all of our peptides together in a single tube. So we 228 peptides, we add our protease of interest. We have a control, which is our inactive protease, and our active protease. We incubate it for a certain amount of time, and the protease will start chopping up these peptides. We use some mass spectrometry, and we detect these cleavage products coming up as new peaks in our mass spec. And then this is just an example of 3 peptides here, where this random protease has cut it between these 2 phenylalanines. It also cut this peptide between the Y and the D, and this peptide between the Y and the L. And so when we look, and of course we're going to get a long list of maybe 100 of these. And when we look at this information here, we can see that there's some reason why the protease cut, let's say, this sequence here, VYLT, at this site, and didn't cut here. So that's what we call the specificity. So obviously the side chains of these amino acids fit nicely in the active site of the protease, which is why cleavage occurred here, here, and here. But what we can do is we can look at the amino acids at either side of these cleavage sites, and we can essentially align them up here. And we call the amino acid in this position here the P1 residue, P2, 3, 4, as we go from right to left. And then we call the amino acids in these positions P1 prime to P4 prime, from left to right. And so we can calculate the frequency of how often amino acids appear in these positions, because that will give us an idea of the types of things that fit well into the active site of the protease. And we can look at, let's say, 100 of a list that's 100 long, and we can come up with these logos, which we call ICE logos. And essentially, the height of the letter, the height of the amino acid, corresponds to how frequently it's found in each position. So this is an example of one of the proteases we worked on, and you can see that this protease really likes tyrosines, phenylalanines, leucines in this position. It really likes arginine in this position. So we can get this kind of a chemical snapshot of what fits well into the active site of the protease. So going back to the question I posed earlier is, can we design a better protease inhibitor? And so how we did that is we took the human proteasome and the Plasmodium proteasome, because we really wanted to compare the difference in their ability to cut this peptide library. We incubated the peptides with the proteasome, and we saw several hundred cleavages, and we saw many peptides that were cleaved in the exact same position between the 2 proteasomes. Some peptides were differentially cleaved, and then some peptides, there was some overlap where you had some overlapping cleavages and differential cleavage. But in total, we had more overlap than unique, but certainly the Plasmodium is different enough from the human proteasome that, that we for the potential to develop some Plasmodium specific peptide inhibitors. We calculated these logos. Again, this is, each of these amino acids of frequency, but I don't really want to spend time on this because there's a lot going on. It's a busy slide. And so what we essentially did, so since every one of these amino acids has a frequency behind it, that we essentially subtracted, looked at the difference in frequency of every amino acid in every position between here and here. And what we're seeing here is that in this heat map is that the more yellow the amino acid is, the more frequently it's cleaved in this position, in a position by the Plasmodium proteasome. So you can see that this tryptophan here in the 3rd position is bright yellow. So what we're basically saying is that the frequency of this W here, tryptophan, is much higher than the frequency of the W here. So this is in subtract. So we found that in particular that tryptophan here, leucine here, and tryptophan in the P1 position were more selective for Plasmodium. So we can take this information, this WLW information, and we can go back to our starting protease inhibitor, which had worked quite well, this trileucine. And we said, well, maybe we can change this leucine to a tryptophan and this leucine to a tryptophan. And in theory, we should shift, the fitting, this inhibitor should fit better in the active site of the Plasmodium and perhaps fit worse in the active site of the human. So we made 3 different inhibitors. We made the WLL version, where we just had the W here. We made the LLW, and then we made the 2 of them, which we call WLW. We tested this against malaria and against human fibroblast cells. And lo and behold, we found that this molecule, was simple change from L to W, increased the potency against malaria by about 12-fold, slightly increased the potency against human fold. But we were very excited to see that we had now made an inhibitor that was 242-fold more potent at killing Plasmodium than fibroblast cells in a 72-hour assay. So we went away and we injected, we infected 12 mice with Plasmodium. This is the Plasmodium burgii species that mice can get infected with. And you can see in 6 of the mice, we infected on day 0. After day 6, half the mice, 6 of the mice had already had percent parasitemia was about 28%. And essentially what that means is that 28% of their red blood cells had detectable parasites. You can see these parasites here, these distinct punctate staining in the red blood cells. However, for the other 6 mice, we did a single injection on day 3 of this compound, this orange compound here, and we saw complete, complete reduction in parasitemia. So no growth in parasitemia from a single injection of this, and no notable toxicity to these mice. We solved the cryo-EM structure with our collaborators at the University of Cambridge. And I won't go into detail here, but we saw some good 3.4 angstrom resolution of the Plasmodium proteasome with Coetzee inhibitor bound inside. And we discovered that this inhibitor bound to the Plasmodium proteasome in both the beta 5 and the beta 2 subunits. So we were able to develop these really potent inhibitors of these subunits. And so we continued to work in this, but I began to collaborate in San Diego with a professor here at the Institute of Oceanography. His name is Bill Gerwick. And Bill goes out to the Caribbean and he samples, he's a chemist, and he samples, he's identifying marine organisms and marine microbes for looking for new chemical matter. And so one time he was doing a snorkeling trip in Curacao. So this is a little island north of Venezuela. And on the south coast of Curacao, they did a snorkeling trip. And this is from his grad student's notebook here, that on an afternoon snorkeling in Carnaby Bay, between the Hilton and Marriott Hotel, using an anchor rope, they isolated these brown tufts from an anchor rope, and they call it sample number 1922. They did some 16S RNA sequencing, and they found that it was a marine cyanobacteria within the Symphoca species. They did some chemical extraction, the HPLC separation, and they found that one fraction within this bacteria could kill both human cells and Plasmodium cells. They were able to isolate it, and they call this compound carmaphycin, basically karma after the location, carmaphycin B, and is a peptide that has this reactive epoxy ketone group and this lipid group. When they tested against parasites and human cells, they found it was very potent against parasites. So again, the lower the number, the better here. But it was also very cytotoxic against human cells. So it had a very low selectivity index, and they discovered that it was inhibiting the beta 5 subunit. And so the medicinal chemists in the lab then began to make some modifications of these that were decreasing the cytotoxicity, so increasing their selectivity. They made a bunch of PEP inhibitors. They didn't really get past this 30-fold mark until they began to change the stereochemistry of this amino acid here from an L amino acid, which is naturally found, to a D amino acid. And they were able to maintain the potency against Plasmodium, but greatly increase the cytotoxicity to 300-fold. And just when we thought we had a feel for what we knew was going on, we began to make more compounds like this one here with the pyridine alanine, and realized that even some subtle changes basically make this compound over 2,000-fold or maybe 5,000-fold more potent against human cells. So we completely lost our selectivity. So this was our lead compound here, number 18. We went off and made 60 additional analogs, which I'm not going to work my way through that. So we're up to analog 18 right now, but this analog is a wonderful compound and inhibits both the beta 5 and the beta 2 of Plasmodium. Super potent against malaria at 9 nanomolar and not at all toxic. So it takes 24,000 nanomolar, 24 micromolar to kill a human cell. So we have this selectivity at 2,600-fold. We just submitted this paper is currently under review describing this inhibitor. It's up on bioarchives if you want to see it. It's been there for about 2 months. But because we've shown that we can make such potent and selective inhibitors that are not cytotoxic to human cells, we've recently gotten funded from the Bill and Melinda Gates Foundation and from the Department of Defense, because the US government is always concerned about their soldiers getting malaria. So we got some funding to really try to make this into a more stable drug that can be taken orally, because currently it's not particularly well, it's not orally bioavailable. And so in the last two slides, I wanted to show that, hopefully I've convinced you that, we're pretty good at making inhibitors that are super potent against malaria and against the anti-target, which is us humans. They are not cytotoxic at all against humans and mammalians and rodents. And that brings me back to this point here, because all these other parasites that I work with, they all have the same anti-target. So all of these parasites, we want to develop drugs that don't inhibit the human, but inhibit these parasites. So the molecules that we're making, we then screen against these other parasites and find ones that, are good hits. And then we begin to do some medicinal chemistry to make them more potent against these parasites. And this is just a couple of examples here of some molecules that we've developed against organisms called Babesia microti, another one against a worm parasite called Schistosoma mansoni. We have one against the same molecule is very good against Trichomonas vaginalis, which is a sexually transmitted disease. And then we have some projects ongoing in the lab where we've made some new molecules against this brain-eating amoeba called Naegleria fowleri. And we're also developing proteasome inhibitors against Candida albicans. So in summary from my talk today, hopefully I've convinced you that parasite proteasomes are a great drug target. We use this peptide library called multiplex substrate profiling by mass spectrometry, or MSPMS, to rationally design our peptide inhibitors. And then we use the data from this to design inhibitors and improve our inhibitors to be more parasite selective. And then the medicinal chemists came in and increased the potency and the selectivity of these molecules. So with that, I want to thank you for your attention. This is just a picture on a very rare cloudy day in San Diego. This is my group here from earlier this year. And so I want to thank, these are my postdocs and grad students, and then these are my collaborators here who are many of the parasitologists. So thank you for your time.
Thank you very much, Dr. Anthony O'Donoghue. It was a very nice presentation. It's a topic that I really like. And I really appreciate the overview of your work. Thank you for the audience that I can see they are posting questions here. We are using the questions at the end of the presentation as the presentations, as I said, for the discussion. So let's move to our second speaker, which is a friend of mine, is Dr. Nicola Calvani. She will talk a little bit about her experience of working in Southeast Asia as a parasitologist. Her talk is Phaceula Species in Southeast Asia, a livestock parasite of zoonotic concern. Dr. Nicola is currently a postdoc at the Molecular Parasitology Laboratory at the University of Galway in Ireland, where she works with Professor John Dalton and his team on parasite protein characterization and development of vaccines against the parasite Phaceula hepatica in sheep. Before moving here, Dr. Nicola completed her PhD in molecular diagnosis and species differentiation of Phaceula species in livestock in Southeast Asia and at the University of Sydney in Australia. Dr. Nicola also recently developed or created or launched the social media named Hermitology, where she actually works to profile women in parasitology, which is a very nice work. So welcome, Dr. Nicola, and now it's you.
Thank you, Carolina. Let me just share my screen now. Is that visible? Are we good to go? Yes? Okay, brilliant. Okay, so I'm going to talk to you today, as Carolina said, about liver flukes. So that's a species that we might be familiar here in Ireland that affects the sheep and sometimes also the cattle that we have here. But it's actually a global parasite of concern. And as Carolina mentioned, I've spent my PhD and early research career working on it in both Australia and Southeast Asia. So before I get into a little bit more detail, I'm just going to briefly outline the life cycle for you. It is an obligate two-host parasite, which means to complete its entire life cycle, it has to first go through an aquatic snail host, from which a larval form of the parasite emerges and exists on vegetation at or near the surface of the water, and that is the stage that is infective to mammals. Mammals then come along and eat these stages, and that includes in livestock, sheep, cattle, buffalo in Southeast Asia, but also people. And once these infective stages are ingested, they emerge in the intestines and penetrate the intestinal wall, migrate through the abdomen to the liver, and then they spend the next sort of 8 to 12 weeks growing and moving around the liver, where they cause significant pathology. After that point, they move to the bile ducts, where they're sexually mature and they reproduce and lay eggs. And these eggs are shed back out into the environment. A different larval form of this the stage of the parasite then reinfects these snails, and the cycle then continues. In Southeast Asia, where we have aquatic rice production ubiquitous throughout most of the region, we have a perfect storm for this parasite, where we're seeing a lot of these aquatic snails interacting with livestock, either through the draft and ploughing processes of the fields, or where we're using their manure to fertilize the fields. And then we also have people that grow vegetation and food and salads that they then consume raw. So we have a really great system in place to maintain this life cycle. It is a significant parasite of people in a lot of the world. Over 180 million people, mostly women and children, are considered at risk of infection, and therefore the World Health Organization has actually designated it a neglected tropical disease. And it is a significant cause of livestock production losses globally. There are two main species, Fasciola hepatica, which is the one we have here in Ireland and the one we have back home in Australia, and it's got a temperate distribution. And then we have Fasciola gigantica, which is more tropical, and it's found in many low and middle income countries in Asia and in Africa. It is a truly global disease. You can see here, this is countries where human cases have been reported. It's found in over 70 countries and on every continent except Antarctica. But there are also definite regions where we consider it to be hyperendemic in the human population, which means that we have a focal point of infections that occur above a standard that you would expect to see. One of those regions is in Vietnam, which you can see on the map here, where over 20,000 human cases have been reported. However, I am personally of the opinion that this is a bit of a selection bias. So there's not to say that there's not these hyperendemic regions also occurring elsewhere in Asia. It's just that we haven't necessarily been looking for them. So the human and livestock cohabitation and production and agricultural conditions in these regions are similar. So it doesn't necessarily mean that because we haven't detected it in neighboring countries to Vietnam, such as in Laos and Cambodia, that it's not occurring. The distribution of these two species was traditionally associated with post-domestication of livestock movements. So Fasciola hepatica spread out through the Middle East into Europe and into the Americas and then into Australia. And then Fasciola gigantica spread from sort of the lowlands of Africa up through to the Middle East and then through into Asia. But we essentially assumed that that was the distribution of this parasite and then forgot about it. However, I'm sure you're all aware that international livestock trade is occurring more frequently now in a higher volume than ever before in human history. And so basically, the theory is that